Hello and welcome back to Teach Me in 10, the video series that's brought to you by LabTube, which is part of the Technology Networks group. And here at Teach Me in 10, we are making it easier than ever to access complex areas of science and to allow you to learn something new. It's where we challenge scientists to present and summarize their research or scientific concept in less than 10 minutes. So before we begin, my name's Lucy and I'm a scientific content producer here at Technology Networks. And today I'll be speaking with Shabana Islam, a product line manager, and Roxana Ghadessy, the digital manager for commercial excellence, both from Corning Life Sciences. We'll be speaking about air-liquid interface systems, and air-liquid interface, or ALI, is ideal for studying respiratory tract epithelial cells because it exposes one side of the culture to liquid media and surrounds the other with air. And because of this, ALI systems basically allow researchers to much more accurately mimic the in vivo conditions compared to using conventional cell culture models, which makes them ideal to perform mechanistic studies of respiratory epithelial cells as drug permeation barriers, to model respiratory diseases like cystic fibrosis and asthma, to investigate essential dermal processes such as wound healing, and to advance our understanding of respiratory epithelial infections, including SARS-CoV-2. So from asthma to COPD, ALI models have been critical in researching respiratory diseases. With the recent outbreak and severity of COVID-19 virus, this technology is becoming even more critical in the race to understand and treat disease. And with the popularity of these much more complex models growing, we've brought together two experts to teach you a bit more about disease modeling using ALI tools and systems. So without further ado, I'll pass over to them both. Hello, how are you both?
Great, thank you. How are you?
Thank you.
Very well, thank you. Fantastic. So we'll jump straight into the first question, which is what is air-liquid interface, or ALI?
Yeah, so air-liquid interface, or ALI, is based on a dual compartment model, which is separated by a microporous membrane that's typically coated with collagen. So the insert with the membrane sits in a base plate, and this establishes a two-compartment model with both apical and basolateral surfaces exposed to air and liquid. Compared to conventional submerged 2D cell culture, the ALI configuration mimics the conditions found in vivo and drives differentiation of the cells. And because of this, ALI culture have become established as a gold standard.
So what applications are ALI models used for?
Well, ALI models are quite versatile and they are suitable for a variety of different applications. Starting with basic research, the technique is ideal to study respiratory tract biology. For example, mechanistic studies of airway epithelial cells as drug permeation barriers. Preclinical research applications include dermal processes. For example, if you are interested in wound healing or skin remodeling. And then finally, from a disease perspective, ALI is commonly used to effectively model respiratory diseases. For example, asthma and cystic fibrosis, including more recently, as we are aware, infectious diseases such as COVID-19.
And what benefits can ALI bring over other models for certain applications?
Yeah, so there are several benefits of ALI, and I will share 3 examples. So let's start with cystic fibrosis. Because of the complexity of this disease, the symptoms can differ from person to person, and so its patient-to-patient treatment response may vary. So in this case, for example, nasal cells collected from patients and cultured with ALI allows researchers to explore personalized approaches. Another great application is a 3D model of human airway epithelium, for example, mucil air, that has been successfully cultured using ALI technique. Wound healing, as Roxana also mentioned, is another great application of ALI model that can enable cultured keratinocytes to differentiate into organotypic skin equivalents and display similar characteristics to the skin in vivo. So as animal models are very expensive and time consuming, and there is considerable animal to animal variation. So with ALI culture, you minimize that variability and can grow cells faster.
Why is ALI so relevant right now, especially during the pandemic?
That's a great question. COVID-19, it is a respiratory disease, and the virus SARS-CoV-2, the virus targets airway epithelial cells. Now, these cells, they can be nicely cultured at the air-liquid interface using permeable support model. And this creates the pseudostratified epithelium that contains the differentiated airway cell types. So this model can be efficiently infected with the coronavirus. So therefore, it is a useful tool to understand perhaps the biology of the virus, how it infects, how it replicates, and also its cytopathic effects. Furthermore, ALI cultures can be coupled with other 3D culture techniques, for example, organoids. And this is if you wish to create a more highly relevant preclinical tool to study SARS-CoV-2 infection, for drug validation, and also for screening applications.
And what tools can be used to support successful ALI models?
Yeah, great question. So permeable support such as Transwell, Falcon, or Corning BioCoat inserts, typically coated with collagen, has been widely used to support successful ALI models. So researchers may choose to self-coat the insert either with collagen type 1, type 4, or a combination of other collagens, or use pre-coated BioCoat inserts, which saves time and labor. Typically, 0.4 micron pore size inserts are used for ALI studies. Regarding the choice of culture wear format, it will depend on what your end goal is. For example, if you are growing 3D organotypic skin equivalent model, larger size formats such as 6-well inserts, 100mm dish, or a large Transwell format would be best suited. Whereas for studies related to drug screening, the 96-well high throughput screening format would be more ideal. And if researchers wish to couple ALI with other 3D cell culture models, such as organoids, extracellular matrices such as Corning material matrix are also useful tools.
And what are some protocols for ALI culture?
There is a general workflow to create ALI cultures, and you may need to optimize this. It really depends on your starting cell type. The workflow itself, it starts with a phase of conventional 2D cell culture. And here you would have your undifferentiated cells. They would be expanded, submerged, typically on a cell culture flask towards confluency. The cells are then transferred. They're seeded onto the apical surface of the collagen-coated membrane, and growth media would be added here in both apical and also the basolateral chambers. So then once a confluent monolayer has formed, the growth media is removed, and differentiation media is now added only to the basolateral chamber. So at this point, we are exposing the apical surface of the cells to air. We are essentially lifting the culture, and this creates that air-liquid interface condition that drives the cellular differentiation process. At which point you would routinely change the media in the basolateral chamber, and after several weeks, the cells would fully differentiate and form this in vivo-like pseudostratified epithelium layer.
And do you have any tips to consider when you're using permeable supports for ALI culture?
Well, there are several tips which you can consider, and I will just mention a few here. To start with, it is important to optimize your initial cell seeding density onto the permeable support membrane. And this is to ensure good cell attachment. A second tip that you can consider is the use of the Falcon deep well plates, particularly during the longer term differentiation phase. And this can help you to reduce the frequency of the media changes to save time, and importantly, minimize the contamination risk. A final tip is it is important to keep the cells, the surface of the cells exposed to air. So you should carefully monitor your ALI cultures. And if you observe any residual liquid or any excess mucus which is being secreted by the cells, then you can use a pipette tip and you can gently aspirate the liquid at the edge of the membrane.
And then our final question, as we're almost out of time, but what resources can you share that are helpful for ALI model support?
Yeah, so we have a dedicated page on disease modeling at the Air Liquid Interface at Corning website, where we have a broad section of literatures available, such as airway models, skin models, citation summary, as well as application note on COVID-19 research. And we also have protocols on ALI generation using different models, uses and best practices for ALI systems, tissue engineering, as well as the nucleus blog page. And finally, we also have pre-recorded Bright Talk webinars available for you.
Fantastic. Well, I think that was a fantastic way to kind of end this Teach Me in 10. So thank you very much for joining me here today, both of you.
Thank you
so much.
It's been really enjoyable. Thank you.
What a fantastic episode. I quickly want to take this chance to thank Roxy and Shabana for joining me here on Teach Me in 10 today. It was a fantastic episode. We'll be back next week for another installment of Teach Me in 10, but if you just can't wait till then, we've linked the hub page down below where you can see all of the videos that we've published so far.